Hello everyone, and welcome to the second half of Chapter 5. We're going to be talking about proteins and nucleic acids. So, proteins have different functions, like receptors, structure, storage, and enzymes. So what makes a protein? Well, we have amino acids, which are the monomers, which will make peptides, and then peptides are used to make proteins. There are 20 different types of amino acids, and those are what are going to dictate what a protein will do, because structure determines function. So a deeper look at amino acids. So we will usually have a central carbon, which is bonded to an amino group on the left and a carboxyl group on the right. It will also have a hydrogen bonded to it and a R group. So in making a polymer bond or peptides, so we will use dehydration synthesis between two different amino acids, as shown here. We will take the hydroxide from the carboxyl group and the hydrogen from the amino group. Use dehydration synthesis to make water, and it will be removed, and then a peptide link will be made between the carbon and the nitrogen. We do this many times over to get a polypeptide. So there are two different shapes for proteins: globular proteins, which are circular, and fibrous proteins, which are usually more straight looking. There are 4 levels of protein structure: primary, secondary, tertiary, and quaternary. And we're going to go more depth on those. So we have primary structure. This is when you make a polypeptide, and as previously said, it is when you join different amino acids together. A very important key about this is that there will always be an amino end and a carboxyl end. So if an amino end would be over here, a carboxyl end will be here. Next is the secondary structure. This is made by covalent bonds between the different molecules inside of the polypeptide, as shown over here. For the A helix, it will be more circular, and we will have hydrogen bonded to oxygen. But for the B plated sheet, it will be more rigid, and it will usually have 2 lined up like this, where they will have a hydrogen bonding to make sure that the structure is stable. The tertiary structure, that is the 3D structure of the polypeptide, and this is determined usually by the R group. So we have 2 types listed over here. So we have hydrophobic interaction, and that is when there's water on the outside, and the amino acid will interact with it and immediately go to the center, where it will be held together by van der Waals forces. And the other is disulfide bridge. This is made by 2 different sulfurs that are bonded to each other. And then there's the quaternary structure. This is made by having multiple tertiary structures, and the tertiary structures become subunits, which are part of the overall whole protein. So there's denaturation and chaperone proteins. So denaturation is when a folded protein gets unfolded, and this can happen because of pH or the temperature is not right for the protein. And chaperone proteins are what help fold proteins in the right way. So sometimes proteins are mistakenly folded, or they haven't been folded yet, and so chaperones will help fold them properly. Now we have nucleic acids. So there are two types of nucleotides, DNA and RNA. So what makes a nucleotide? Well, nucleotides are made by a nitrogenous base, a sugar, and a phosphate group. So let's look at the nitrogenous base. There are 5 different nitrogen bases: adenine, guanine, cytosine, uracil, and thymine. Adenine and guanine are purines, while cytosine, uracil, and thymine are pyrimidines. We notice over here that the purines have a six-membered ring with carbon and nitrogen, with a 5-membered ring, while over here with the purines we only have a 6-membered ring with carbons and nitrogens. Now for this sugar group, there's ribose and deoxyribose. This is mainly differentiated by the oxygen bond, where the ribose will have this oxygen, but the deoxyribose will not have the oxygen. Another important thing is when naming the sugar carbons. So for example, this will not be 1, it will be 1 prime, and this is a way to differentiate the carbon when numbering the carbons and the overall structure to make sure that the carbons and the nitrogenous are just normal numbers, and then in the sugar they will have a prime after numbering. So we have a phosphodiester bond, and this is made by bonding 2 different nucleic acids. This bond happens by a phosphate, and it will bond to a 3' end. It will bond a 3' end and a 5' end. And this will be bonding the amino acids, or this will be bonding between the 2 sugars, and this will make a phosphate sugar backbone, but the nitrogenous bases will not make that backbone. And then let's go back to DNA and RNA. So over here, we have DNA, which is a double helix, and when pairing, usually we'll have a 3' end running next to a 5' end. So they're not parallel, they're actually anti-parallel, and this makes sure that everything's complementary. So when having the different nitrogenous bases aligned to each other, adenine will always pair with thymine, and cytosine will always pair with guanine. In RNA, uracil will be substituted for thymine, so adenine will be pairing with uracil instead of thymine, and it is a single strand. That's it.